All right, it looks like we're ready to begin. So let's get started. So hi again and welcome to today's webinar. Just a quick intro. My name is Yuan Tian and I am the Content Marketing Manager at Biocellular Boston Corporation. I'll be the moderator for today's session. We are really excited to bring you this webinar focused on CAR T cell therapy, which is a topic that continues to generate so much innovation and discussion in the field. And that said, we are honored to be joined by two speakers today, Dr. Marco Ruella and Dr. Fengqian Chen. So before we hear directly from them, I would like to start with a quick introduction of each speaker, just to give you some background on their expertise and their perspectives. So first up is my honor to introduce Dr. Marco Ruella. So Dr. Ruella is a highly respected leader in the field of CAR T cell therapy. He earned his medical degree with high honors and completed his specialization in clinical hematology at the University of Torino in Italy. He was attending physician at the Hematology and Cell Therapy Division of the Mauriziano Hospital and was an instructor at the Biotechnology School at the University of Torino. So from late 2012, he was a postdoc fellow and then an instructor at the University of Pennsylvania in the Center for Cellular Immunotherapies, working in the laboratories of Dr. Carjun and Dr. Sargil. And from 2017 to 2018, he served as Associate Director of Dr. Jun's laboratory. And in 2018, Dr. Ruella was appointed Assistant Professor of Medicine in the Division of Hematology Oncology and the Center for Cellular Immunotherapies, as well as the Scientific Director of the Lymphoma Program at the Hospital of the University of Pennsylvania. And Dr. Ruella has been recognized with numerous awards and honors. There are far too many to list today, but let me just mention a few highlights. He received the inaugural SITCEMD Serono Cancer Immunotherapy Clinical Fellowship, the AACR-BMS Oncology Fellowship, the ASH Scholar Award, and NIH K99R00 Award. He has also received the Paula Kamapis Award for Leukemia Research, the Gilead Sciences Research Scholar Award, and the Gabriel's Angel Foundation Award, among many others. In addition, Dr. Ruella is the author of many peer-reviewed publications on targeted immunotherapies for hematological cancers and is also an inventor in several patents on CAR T therapy, as well as a scientific founder of Victoria Biotherapeutics. So thank you so much for joining us today, Dr. Ruella. We are really honored to have you with us today, and we look forward to hearing your insights. And
thank you for having me.
Thanks. And next up, it's my pleasure to introduce Dr. Fengqian Chen. So Dr. Chen earned her PhD at Texas Tech University and the University of North Carolina at Chapel Hill School of Pharmacy, which is a top-ranked pharmacy school in the US. She spent 4 years conducting doctoral research in the lab of Professor Li Huang, who is a world-renowned pioneer in lipid nanoparticle or LNP technology. And following her postdoc training at the University of Maryland School of Medicine, Dr. Chen led a biotech cell therapy team for 3 years. And with more than a decade of preclinical research experience, she has contributed 38 US patents and publications in the fields of cell therapy and cancer immunotherapy. She also serves as editors and reviewers for high-impact journals in cell therapy and LNP research. Having previously collaborated with Biocellular as a client while leading a cell therapy team, Dr. Chen joined Biocellular in 2024 after 2 years of successful partnership with Biocellular. She now serves as regional director at Biocellular and drives innovation and advances therapeutic discoveries with her expertise in humanized mice. So welcome, Dr. Chen, and thanks so much for joining us today.
Hello, everyone. Thank you for having me.
All right. So now just to quickly go over today's agenda, first, we will hear from Dr. Marco Ruella, who will discuss how cancer cells manage to evade CAR T cell targeting and the innovative strategies to overcome that resistance. And after that, Dr. Fengqian Chen will talk about how target humanized mice can be applied in CAR T development. With that, just a quick note before we begin. This webinar is being recorded. and we will have time for Q&A at the very end. So if you have any questions along the way, feel free to drop them into the Q&A box. We will address them after the presentations. Okay, now without further delay, let me hand it over to Dr. Ruella.
Well, thank you. Thank you so much for the kind introduction. Way too long and too generous, but I appreciate that. And thank you for Biocytogen to organizing this webinar. So, of course, the topic is very broad. And so we want to talk about resistance to CAR T-cell therapy. So what I'll try to do today is to give you an overview of what we know so far about resistance to CAR T-cell therapy, and then maybe focusing on a couple of projects that we are running in the lab that I think can be interesting for this audience. These are my disclosures that might be relevant for my presentation. And so I think that there is a lot of interest in CAR T-cell therapy because since 2017, when tisagenlecleucel, a 4-1BB co-stimulated CAR T-cell product, was approved by the FDA for pediatric and young adult ALL, we've had a lot of products that have been approved. And so Tisa-cel was initially developed at Penn and then was licensed to Novartis that brought it to approval. And since then, we have had a lot of products that have been approved: Axi-cel, Brexcel, Liso-cel. We have now 2 products for multiple myeloma targeting BCMA. And then most recently, we also had a new product for relapsed refractory BALL. So we have overall 7 products approved in the US for both lymphoma and myeloma. And we have 2 targets, CD19 and BCMA. But obviously, even with this all excitement that we see and we share for CAR T-cell, we need to recognize that at long term, even in the best case scenario for adult patient, which is lymphoma and CAR-19, you can see that we are able to cure about 30% of the patients. This is a 5-year follow-up that you can see here of a trial of Dr. Schuster at Penn. And in diffuse large B-cell lymphoma, 31% of patients were without disease. So this is remarkable because we can say that probably we cured about 1/3 of patients, but at the same time, we're not curing 70% of patients. Likewise, there are toxicities. We know that after infusion of CAR T-cell, you can have cytokine release syndrome. Then there can be neurotoxicity. There are cytopenias. There are the so-called HLH, that is a macrophage activation syndrome. So there are a lot of toxicities, a lot of causes of relapse that we need to study. And so the mission of my team is really the one of studying the resistant mechanism, the toxicity mechanism, so that I can then develop with my team novel effective approaches. And so what we have learned so far, and I try to keep this slide up to date. It's a complicated slide, but I will guide you through it. So this slide tries to summarize what is known at this point about resistance to CAR T-cell therapy. And you can see that I always want to start with the so-called pre-infusion barriers. So this means that CAR T-cell is a quite sophisticated, expensive therapy that might face issues even before you administer it. What do I mean? Well, I mean that the high cost and the access can be a problem. There could be progression during manufacturing. There might be some patients that don't have enough lymphocytes, and so you cannot make the product. And so I like to consider these factors also factors that affect the success of CAR T-cell. And then host factors are becoming sort of a new area of interest, where the idea is that there are multiple factors like the tumor burden, potentially immune rejection, microbiome, that can affect CAR T-cell functionality, but they're not really studied that much. And so today we will focus on these topics. And why we are interested in this topic? Well, because they can affect all of these other functions that I listed here. CAR T cell dysfunction is a big issue for CAR T cell. If you have exhausted T cell or terminal differentiated T cells, we know that they're not going to be as effective as we would like to against tumor cells. The tumor microenvironment represents a major barrier to the success of CAR T cell therapy, especially in solid cancers. And lastly, the tumor itself can become resistant to CAR T cell. For example, one of the most common features is the loss of CD19. And so now the tumor is basically invisible to the CAR. So of course, we're not going to be able to go through all of these mechanisms. Again, today I want to focus a little bit more on the role of the gut microbiome and the role of potentially other host factors that can affect CAR T cell function. So let me show you first one piece of information that has been published. So you can see more details in this collaboration with Dr. Van der Brink and Dr. Smith when they were both at Memorial Sloan Kettering. And so the hypothesis here was that the gut microbiome, which means that the bacteria that we have in our gut, can affect the efficacy, but also the safety of CAR T cell products. And so to study that, we used two cohorts of patients. One cohort was the cohort of patients where we look at the antibiotic use before CAR. And why is it important to look at antibiotics? Well, obviously, if you take antibiotics, they're not going to only cure your infection, but they're also going to affect the bacteria in your gut. And another cohort of patients, with about 50 patients, where we studied the fecal microbiome, the gut microbiome in these patients. So we took stool samples before treatment and we analyzed the composition. First interesting piece of information. Our patients take a lot of antibiotics. You can see that overall, about 65 to 70% of patients would have taken at least one antibiotic before CAR T cell therapy, in the 4 weeks before CAR T cell therapy. And some of them are sort of prophylactic antibiotic, like Bactrim. But we do have antibiotics that are given for neutropenic fever, for example. One of them is piperacillin tazobactam, Pip/Tazo. For example, we have cefepime. And then there are also other broad-spectrum antibiotics, for example, meropenem or imipenem, and so on. And so we call this category PIM. So these are all the yellow ones that are known to be broad-acting antibiotics that might have an effect on the composition of the microbiome. And so that's what we define this category called PIM, Pip/Tazo, imipenem, cilastatin, and meropenem. Okay, so what happens if we go and look at our patients and we just look at the overall survival, for example, of patient exposed or not exposed to antibiotics? Well, the data were pretty remarkable because you can see that patients in yellow who were exposed to antibiotics before CAR T cell therapy, so in the 4 weeks before CAR, had a very poor overall survival as compared to patients who did not. Obviously, the question was, well, maybe we're just selecting patients that have more aggressive disease, they are more complicated, and so it's not really the antibiotic, but it's just all the patient characteristics that come with it. Well, to address that question, we perform a multivariate analysis that you can see here. First, we did a univariate and then a multivariate. And we tried to put all the factors that have been associated with poor progression-free and overall survival. You can see, for example, the tumor burden in terms of LDH, the type of product, number of lines of therapy, performance stages, type of disease, age, sex, and so on. And you can see that exposure to antibiotics was an independent factor at the multivariate analysis, really suggesting that potentially it does cause an effect on patient outcomes. And so, of course, we wanted to dive deeper, and we look at the composition of the gut microbiome in our patients. We basically collected the baseline microbiome samples before day 0, which is the day of infusion of the CAR. Very interesting data, because you can see that each column is a different patient. And you can see that there is quite a bit of heterogeneity. For example, these patients here have a lot of lactobacillus. Other patients in orange have a lot of ruminococcus. Some patient might have more bacteroides, and so on. So pretty heterogeneous. So the first question, how are these patients different than healthy volunteers? And so the first thing we look at is the alpha diversity. And you can see that the diversity in patients with that were going to be treated with CAR, it's lower than patients, healthy volunteers. Furthermore, the so-called beta diversity was higher. So there was more heterogeneity within these samples as compared to healthy volunteers. And you can see that if you do a principal component analysis, you can see that the healthy sort of cluster here to the right and the CAR T cells. So they are very different than healthy population. And this is probably due to the antibiotic, to the previous treatments, and so on. But of course, the big question is, can we find some groups of bacteria, some genera, for example, of bacteria or species that can be associated with a better outcome? And you can see that we saw some taxa that, for example, Clostridia, and in particular the genus of Ruminococcus or Faecalibacterium, but also the Faecalibacterium prosnitzi, that were actually associated with better overall response at day 100. So they suggest that if you have a different composition in your gut, that might correlate with outcomes. And in order to prove that, we selected 4 taxa, actually 5 taxa that have been studied extensively in the setting of checkpoint inhibition. You can see them here. And we asked the question, which one of these taxa associates with better complete response rate at day 100? And interestingly, you can see that if you have high Ruminococcus, you have higher chances of complete response at day 100 as compared to having low Ruminococcus. It's interesting to see that conversely, if you have high bacteroides, you can see here in blue, you have more toxicity. So now you have something that is associated with more toxicity. Obviously, these are trends. It's a small population, but we started to see some actual result here. And so we were the first to publish that, but there were other papers that were published about this topic. And you can see that a team from China showed some of the same, for example, cohorts of Ruminococcus, Faecalibacterium, for example, that have been associated also in our case with better complete response. Another group from the US and Germany also had a similar trend with Ruminococcus. So clearly there is a correlation between the gut microbiome and the outcomes. So taking that in mind, we wanted to take this concept to the next level. And this is something that is unpublished, and we presented some of this data last year at the American Society of Hematology during the plenary session. And the question here in this work, that is a collaboration with Dr. Mayan Levy, who used to be at Penn, now is in Stanford. The question was, can we give different diets to the mice? And by doing that, can we affect the way CAR T-cells work? Initially it was mostly focused on the effect on the microbiome, but then it became a broader project talking about how the metabolism of patient is affected, in this case of mice, and I'll show you patient data as well. And so we use different diets. One is a controlled diet. The other is a high fiber diet, then high protein, then a Western diet, which is basically a high fat with cholesterol. And then we use a ketogenic diet. So ketogenic diet is mostly fat with a good amount of protein. And so, again, I want to stress the importance of modeling this in animal models. So we use an immunocompetent bulb C model, and the A20 lymphoma cell line. And you can see here the design of the animal experiment. Bulb C host, A20 lymphoma cell line sub-Q. In the meanwhile, the mice will get the diet, and we do lymphodepletion. Again, it's immunocompetent mouse model, so you do need lymphodepletion. And then we will give either CAR19 or untransduced T cell. And the results were very, very interesting. I mean, personally, I was sort of rooting for the high fiber because, I think that we are taught that should be the best diet for our health. And probably is the case, and maybe we should try more type of fibers. But in this specific experiment, really the diet that showed the best outcome was the ketogenic diet. So ketogenic diet, as a reminder, avoiding carbohydrates, mostly focus on meat, high protein, fat, mostly avoiding anything with carbohydrates. And okay, so what happened to these mice when they are exposed to a ketogenic diet? Well, what happened with a ketogenic diet? So you don't have a lot of glucose, and so the bodies start using ketone bodies that are the results of beta oxidation in the liver. And so ketone bodies become the main source of fuel for our T cell. And indeed, when we did metabolomics, we found that beta hydroxybutyrate, so this is called also BHB, the abbreviation, was the top enriched metabolite. You can see it's the one that is to the right, to the most right. Was the one that was enriched in mice getting the keto diet. And so the question was, well, maybe we don't need to change completely the diet. We can only give the beta hydroxybutyrate. And indeed, that's what we saw. We saw that if you give the beta hydroxybutyrate, you actually can get a very nice anti-tumor effect that is as good as the ketogenic diet. So what happened in these mice? Well, what was interesting is to see that there is a big expansion of the CAR T cell in the tumor. So you can see both with the BHB and the ketogenic diet, you see this expansion of the CAR T cell in the tumor. And so the next step, again, you can see the progress in using animal models that are closer to humans. We use an NSG mouse model. So this is an immunodeficient model where we engrafted a lymphoma cell line, OCLY18, and we give the diet through a gavage. In this case, we actually only gave the metabolite, BHB, not the ketogenic diet, only the ketone body. And then we gave human CAR19 or control T cell. And the results were pretty remarkable from my point of view, because if you give the metabolite by itself, you don't really see a major change in the outcomes here. But when you give CAR19, most of the mice will relapse. When you give CAR19 plus BHB, you can see that most of the mice are actually in complete response, really telling you that BHB can push the function of the CAR. So what's happening? When we measure the expansion of this CAR T cell in the peripheral blood, what you can see here, that you can start seeing expansion of the CAR T cell in the peripheral blood only when you give BHB. And these CAR T cells are actually pretty active because you will find human interferon gamma in the serum of these mice. And why is that? Well, the reason is that the metabolism of this CAR T cell is completely different. So you will see in blue, these are the CAR T cell without BHB, and in red, these are the CAR T cell with BHB. And this is so-called a seahorse assay, which basically measures the oxygen consumption rate in different situations. And so you can see that the maximal oxygen consumption is increased as compared to control. So they are able to get a lot of oxygen and use it, which is good in the setting of low oxygen, like in the tumor. They're able to use more oxygen. And the spare respiratory capacity, so their ability to, as you can see here, is also increased. So what's happening to these T cells? Why they are showing this enhanced metabolism? Well, we tried to understand what was happening by using radioactive glucose and radioactive BHP. And we tried to ask the question, which one of these two metabolites is preferentially taken up by the cell in the Krebs cycle? And the results were pretty dramatic, because you can see that BHP is preferentially incorporated in the mediators of the TCA cycle, while glucose mostly goes to lactic acid, suggesting that we're really providing new fuel for our CAR T cell. It's something that, again, continuing this sort of the progress into better models, we went from immunocompetent to humanized, and now we're going to, well, I would say xenograft, and now we're getting into healthy volunteers. So actually human that are getting the supplement. And so the reason why we could do this trial that was approved by our IRB was that we were using a supplement that is commercially available that is called Ketone IQ. And we will give to the healthy volunteer and then measure the function of the T cells an hour and a half later. And you can see that the results were pretty much in line with what we saw with our preclinical work. After an hour and a half, the oxygen consumption rate was much higher after receiving BHP. And likewise, if you look at the mitochondrial ATP production, was much higher after exposure to BHP. So very exciting results that now we are translating to the clinic. We have a trial where, and again, so this is the fourth step, actual patients, where we are only patient receiving commercial CAR-19 with lymphoma. We're giving BHB, and we're looking at of course, efficacy, but most importantly, correlative studies. How does the microbiome changes, CAR expansion? All right, so in conclusion, I think that there is a new field of investigation in the setting of CAR T cell that involves the role of the microbiome. And so from one side, the microbiome that you can see here can affect how the CAR T cell work. And we pointed out, for example, ruminococcus to be associated with better outcomes. But on the other side, we can hypothesize different diet, and now I should add here ketogenic diet, can affect the functionality of our CAR T cell by modulating potentially their metabolism, or potentially through the microbiome and other mechanisms that we are now investigating. So in conclusion, I think that we show that there is an association between the microbiome and the CAR T cell function. It seems like that these antibiotics are pretty bad. So I would say that we're not recommending yet to avoid them, but obviously we are trying to be very careful in the clinic when we need to use this antibiotic or we don't need to. And lastly, I think we're starting to see that some bacteria might be beneficial for CAR function. And in the second part of the presentation, I told you about the role of beta-hydroxybutyrate. We think is really pushing the cell metabolism to the Krebs cycle and OXPHOS. And of course, we're going to have more data during the pilot trial. So all of this work was possible thanks to my lab, and I really want to thank them. You can see the members. It's probably not super up to date, but I want really to thank. I want to thank. Well, for the first paper, Guido, and I want to thank the BHP group with Shen and Ranjani and many others in the lab that now Jason that are doing this work. And of course, my collaborators at Memorial, Mayan Levy, that is not here, but should be the first one because she's my co-senior author on this project, and we're working fully together. And of course, Karjun, the lymphoma program. And thank you to all of you. I see we have many people connected, so thank you for your attention. I'm happy to take questions later.
Thank you, Dr. Ruella. We're going to have the Q&A session after Fengqian's presentation. So yeah, while we are waiting for Fengqian to pull up her slides, feel free to go ahead and enter any questions you already have into the Q&A box. we will be holding the Q&A section right after this presentation. Go ahead, Wenqin.
Thank you. Yeah, very fantastic talk, Marco. So really appreciate your share your real world application use your CAR CD19 in some clinical data, and also congratulations on the Marco just get promoted for the associate professor at the UPenn. So very a lot of a great achievement. So here my section is gonna discovering how the humanized mice are driving the CAR-T discovery. So I'm a phone region director and biocynology. Here, let's go very highly review of those all the CAR-T field from. So Michael just share some clinical patient data. So let me from the marketing analysis point. Let's look at the data. Just recently update in August that we can see that in the CAR-T field, oncology is dominate. Now have a 13 CAR-T therapy has been approved. So majority is in the US and in China. So you can say they are all liquid cancer or blood cancer. And the target is majority is in the CD19 field and also BCMA. So they are all the approved CAR-T therapeutic. I have a list here. And also you can see we create a chat that you can see the global CAR-T market was growing dramatically. Gonna be a project to reach 21 billions by the next 5 years. And also Marco also mentioned that the CAR-T therapeutic cost extremely high. Just because the different manufacturing things. So the CAR-T therapy is still expensive therapy for nowadays. So not in the oncology recent year, so recent 1-2 years, you can see a lot of funding, a lot of research go to the autoimmune disease space. We just highlight some key milestone of a CAR-T for the autoimmune disease. The first patient treated was published in the New England Journal of Medicine. This is traditional autologous CAR-T for the autoimmune treated lupus. And also in the coming year, 3 years later, in the last year, first patient was treated by the auto CAR-T off the shelf. It's published in China. And this year, there has 2 great news share that not only in the lupus field, other autoimmune field like sclerosis has been published in the last 2 months. Treated user auto CAR-T for the autoimmune, and also not only traditional CAR, and you can see that some in vivo CAR field was announced by Captain Therapeutic. They use in vivo CAR to treat autoimmune disease. So now they haven't has any approval for autoimmune disease, but there has a biotech. Kevin is aiming to get their first CAR-T therapeutic approved for the end of next year or earlier of 27 by treating with lupus. You can see the dramatically funding and research go to the autoimmune space, not only oncology, but also autoimmune. When we talk about CAR-T in this field, there has 2 versions of a CAR. For the traditional CAR, including the auto CAR. So T-cell, you can see the collected from the patient, engineer outside the body, it's ex vivo. So express the CAR and expanded, then re-infusion, do the manufacturing, re-infusion into the patient. It's a traditional CAR. In vivo CAR, which is different, that it's a newer approach. We apply some gene delivery vector, for example, LNP, some AAV virus, some virus-like vector, which carry on some messenger DNA directly inside the patient, engineer educate the T cell without removing or modification the cell into the lab. You can see that those are two different CAR version. So in the traditional CAR, you can say that already approved the concept, already show some successful case in the blood cancer field, and some is ongoing. We have just introduced the autoimmune field. It's in the clinical trial, but still in the solid cancer tumor, this part is still a little bit struggling. So the challenge is because Marco also mentioned a little bit about the first tumor microenvironment. So the solid tumor, they have some their own immune suppressive environment, so inhibit the CAR T function and the antigen, not like a blood cancer like BCMA, CD19. The solid tumor has multiple antigen often expressed in the normal tissue, so this is risky for the untargeted, off-target tumor tox. And also the CAR T, so the cell trafficking, rich migration, they have a struggling to migration into the reach to the tumor site. Also the CAR T dysfunction, this is saying exhausting of a CAR T. The most important is the off-targeting risk. So back to the in vivo CAR, it's a very earlier stage. We don't see any approval of a successful case in vivo CAR. It's very earlier. So there's some challenge still there is targeted delivery. Always the thing, if we can reach to the right immune cells, avoid some off-target effect. And also it's controlling the CAR expression level. And also the persistence inside the body is the challenge. Still, there have a safety concern. The in vivo CAR may induce the CRS, some cytokine release syndrome, and also the off-target. So if you see which one is better, depends on the scenario. So there still has a pro and con between traditional CAR and the in vivo CAR. So in vivo CAR, we have mentioned it's very, very earlier stage, but you can see the MNC, those are multiple big pharma already acquire. They spend more than $6 billion to acquisition of a different biotech. You can see those MNC, including AbbVie, Sanofi, Astellas, AstraZeneca, Gilead. So they have an acquisition, the biotech company. You can see the technology platform. They are majority focused on the different type of vector, lentivirus and LNP. So also the target from blood cancer, only CD19, BCMA, they expand to a different target, CD22, CD20, GPC3, TROP2. So those different target has been chosen by the big pharma. So this is the so far update. It's a huge market potential using in vivo CAR to solve the manufacturing issue. So here is the overall landscape of a CAR T therapeutic. So since today, Marco has mentioned a lot about clinical parts. So my section will focus on the if you do the preclinical stage, how you can choose the right model. So I'm gonna introduce application of a humanized mice in the CAR T. When we mention the humanized mice, I would like to introduce the largest humanized mice company, Biocytogen. So here is a brief introduce Biocytogen. So we have a two branch, Hematologic and Biomice. So you can understanding this, just like Apple, they have a two product, iPhone, iPad. So Hematologic and Biomice is under the umbrella of a Biocytogen. So Hematologic, so how to helpful for the CAR T. If you're looking for the CAR binder, so the Hematologics maybe can help. So we do have a 1 million off-shelf fully humanized antibody for your CAR binder choice. And biomice, you may heard this name before. So it's a global leader in the human gene logging mice, offering the best-in-class humanized mice model. So we already have more than 3,000 off-the-shelf humanized mice under the different background. For example, black 6 immunodeficient mice, and also cellular. If something is a very novel target, we can use a gene editing service to provide the right mouse model for you. So all our off-shelf humanized mice is using our unique gene editing service. And also we can provide CRO service. We can provide use our mouse do the service. So it's a one-stop for support your preclinical research. And we already successful collaboration of all the top 10 MNC big pharma already. So this is the global leader in the human gene logging mice biocycling briefly introduction. So this I have mentioned we have a 3,000 off-shelf humanized mice logging model. Here is a highly summary from the very different indication. What we have mentioned oncology, autoimmune. So you can see we actually replace the gene, mouse gene, replaced by the human gene. We can log in the human gene into the mouse for different application, and every month we update our catalog. So we are happy to send the catalog to you, or you can go to our website, check our catalog. If something, if you're targeting, we already have mouse can support your preclinical research. And also from the mouse to the disease model. So I just use this autoimmune inflammatory disease as example. So we are not only a CRO doing the disease model, we also provide our humanized mice. So those model, different type of model you can see in the screen, we already has a validation data. So we can provide a service in the either our US headquarters Boston site or in our Beijing site. We have been a validation. So we are the leading the humanized mice field. We are also leading the humanized mice in the different disease model field. So this is autoimmune inflammatory metabolized. You can see those in the autoimmune field, lupus or EAE model we already established. It's ready to go for your CAR-T therapy field. Oncology part, so we are well established in the different model. So basically 3 portion synergenic model. So we can log in the human protein target gene into the black 6 BOP6 background, and for the cell line, we also can log in some target gene into the mural cell line. For example, if you use PD-1 expressed mouse and your ejaculation, the PD-L1 MC38, basically you can look at human PD-1, human PD-L1 response in the mouse system. The second is widely used for the CAR-T therapeutic is immunodeficient mouse xenograft model. So we have more than 200 human tumor cellular is ready to use. We have a tumor kinetic data. Already have a validation the model. Last but not least, is widely used in the CAR-T therapeutic field is human immune system reconstitution model. So you can see the two part is a PBMC reconstitution or CD34 reconstitution. So biocycling has use our own humanized our own immunodeficient mice has create a validation data for those two fields. With screening donor, we have a pre-screened donor for the PBMC is ready to use. So let's elaborate more about this field. So here's the application. If you use a mouse model for the in vivo CAR, so there has a two model for you. So first, let's look at wild type mouse. So you can say use this publication as an example. So this in vivo CAR, they use LNP. Is targeted to the mouse CD5. So here is the thing. So if you're in vivo CAR, I want to answer the research question is for target into the human T cell, like human CD5. So those wild type mouse, which express the mouse CD5, not human CD5, will have us translational risk there. So we know FDA earlier this year in April published that they want to reduce the unnecessary animal use. So this part is how translational use wild type to translation to the human. This is research question we want answer. So this is some limitation when we use a wild type mouse. So so if we want to talk about translational, there is two model for nowadays we can choose. Is first, let's talk about on the left side is a human gene logging model, which basically use last page as example. If we log in some human CD5 into the mouse, so this mouse express the human CD5. So if this case, we can look at their LNP distribution in a human expressed CD5 mouse model. So this model is a great thing is, so advantage is it has intact immune system. So it has a results is a reliable and repeatable because basically it's black 6 or BOB6 background just express the human protein. But some disadvantage is so for the tumor study, it still need a mouse tumor. Even the tumor we can engineer to express some human protein, but still it's in the mouse tumor cells, not kill the human tumor. So this is some pro and con use human gene logging. So human second human translation board to the human study is human immune system reconstitution model. There has a PBMC, there has CD34 reconstitution under the immune deficient background. The great thing is we are likely have the human immune system present there, and you can use a human tumor cell. It's great for your CAR-T or cell therapy application. But still, there has a limitation. If for the PBMC, everybody know PBMC is the donor is from the human. It has a huge variation of the different donor. We need a pre-screened donor. Second, for the CD34 reconstitution, so we know the marker is there, so we can see the T cell, B cell is present there. But for example, B cell, myeloid cell, so they pretty likely is premature. Marker is still there, but the cell is not function. So this is still has some disadvantage. Use those kind of human immune system reconstitution model, and the price is expensive as well. So we just here provide the fact to show you different cases. So those two cases, they has the common thing is they will need to apply the gene editing to create the right model for your study. So that when we talk about gene editing, I want to introduce you the biocytogen EGE system. So it's CRISPR-Cas9 based editing system, but we have a technique to advance the CRISPR-Cas9. So this EGE method, we can have a 20-fold higher logging efficacy than normal CRISPR. You can see the data here. If a normal CRISPR-Cas9 technology, if we have 10,000 of mice, only 2 mice can have a successful logging positive mice. But if you use our EGE system, 10,000 mice, you can increase to the 36 mice can have a positive logging. So it's they can have two things help you is first increase just save your time and also save your budget. So that's how biostrategy use this system become the global leader in the human gene logging mice. All our 3,050 mice, we use this technology to help us create the mouse, create those humanized mouse. So this back to the story about in vivo CAR. So we have mentioned that we can log in the mouse, the human gene into the mouse. This is what we did. So this is a mouse, for example, black 6 mouse. So we replace with the human gene. So these are two things we're going to do. First, let's use CD5 as example. We log out the mouse CD5. Second, we log in the human CD5 into that gene. So that is how we create a target humanized mice model. Here, I just show you example here. This is our product, CD3 EDG mice. So the arrow you can show is where we replace with the human gene. So the gene CD3E, CD3D, CD3G was a fully replaced, but those are full protein was fully replaced by the human gene. So there's a 3-layer before we put it into our catalog. We're gonna validation. First, we want to confirm the mouse version CD3 EDG was logged out, and also the human version CD3 EDG is expressed. So we will confirm by gene level, by protein level. Second, because it is for the drug target, we don't only want the protein expressed, we want this receptor, for example, CD3 X function. It need to activate the T cell can be activated. The signal transduction is still intact. This is second layer. Third, even we just change the one protein or one gene, we still want to validate the mouse is still normal mouse. It's still healthy mouse. So this is a 3-layer we're going to do for the validation. All our catalog mouse, 3,500 off-shelf mouse, we will do those things. Let's make sure it's exactly for the drug target. So if you are working on in vivo CAR, your target is a human T. So here is the mouse you may interest in. CD2, CD3, CD5, CD7, CD8 in a different background, BOP6, BLAC6, and some of them is can express in the immune deficient mouse. So let's look at the example for you. So here is this, the publication. You can say the researcher designed the LNP, which is a target for the human CD8. So they want to look at the screening LNP, look at how the LNP can target to the human CD8 in the study their distribution. So this is their research question, want to answer if the LNP can target to the human CD8. How we can do that? So in this case, if this is your research question, I would suggest to use the mouse which express the human CD8. To support your LNP distribution screening. Here's a red mouse for you. So you can see that we actually replaced the human, the mouse CD8A, CD8B gene replaced by the human gene. And also we confirmed by the FACS flow data that human CD8 was only expressed in the homozygous human expressed CD8 mouse, but not in the wild type mouse, because this is a human protein. So we confirm this. And also we want to say something is the CD8 protein, the protein expression in the different immune cell compare wild type and our homozygous CD8 login mouse is identical. The CD8 suppose expressed in the NKT T and CD8 positive T cell. So we can say very identical data. And they shouldn't express in the B, NK, CD4 plus T cell. So this data is very, very identical. So after humanized, this protein is only expressed where they should express, but not in the other place. Also, the last but not least, we want to confirm that the mouse is still normal mouse. You can say that it's from the spleen, the leukocyte subpopulation, different cell type, TB, NK, and also myeloid cell type, and also subpopulation of T compared to Y type, and our humanized CD8 logging mice, their data subpopulation expression level is still identical. So we want this mouse is still normal mouse, still health. So this is a case that if a people target CD8, human CD8, this human CD8 expressed mice is great for your in vivo CAR study. And last model for your CAR-T therapeutic is nowadays widely used in the human immune system reconstitution model. So this model, because we need a reconstitution, use a human cell. So this model is first, there is a huge variation, and also it's a little bit pricey. Then just use human logging system. So we can look at the publication that people has used this, for example, PBMC to screening different CAR do the PK and PD study. Let's look at closely for the this reconstitution model. So PBMC is widely used for CAR-T therapeutic. So you can look at the Biocellular where we made develop this BNDG mice. So we have 350 publication already. So BNDG mice, you can understand, is equivalent to NSG mice. It's lack of T, B, NK cell, and it's similar to NSG. It's on the not background. So, but this BNDG mice provide the cost effectively compared to other immune deficient mice model. So here is so Biocellular, we create BNDG mice, and we support you different project, answer different research question. Is if you are not only look at T cell, you want to look at NK cell, we ate human IL-15 logging to the immune deficient mouse background. And you know, this PBMC, the therapeutic window is very, very narrow because of GVHD. We also log out the B2M, or we also can we also already log out some MHC 1 and 2 mouse to decrease the GVHD. And also we cross the mouse to create, first we can reduce the GVHD, we log out MHC 1 and 2. Also we introduce the IL-15 mouse. So those mice already in our catalog, already in the market. So if you are doing some antibody discovery, IgG therapeutic, so we also have a mouse. It's a double MHC double lockout. And also it's FC receptor lockout, so can reduce your IgG binding to the mouse FC receptor. So we do have a more close to 100 different strain of BNDG. We also can log in, like for example, TROP2. Human TROP2 can express in the immune deficient mice. We also have a different background of a human gene logging on the BNDG, or we can use gene editing to create a mouse for you. So this is PBMC reconstitution. Let me show you the data here. Biocellular, we already pre-screen the donor, PBMC donor, for different strains of immune deficient mouse. So it's in Boston. The donor bank is in Boston, so it's ready to go. You can see those are 3 donors. We test on the BNDG, BNDG B2M, and double lockout mouse. So you can say the PBMC, the one of issue is a huge variation from the donor to donor. That's the reason we need a pre-screened donor, and we have a pre-screened donor ready for you. So you can see double lockout, after double lockout, the mouse survival curve. It's a prolonged to can. We have 12 weeks data, 3 months. So it's pretty good therapeutic window for support your tumor study or other type of research. So besides the PBMC, I want to share that we do have a CD34 stem cell reconstitution. For the autoimmune space, we need a B cell present. probably not B cell function, at least the marker is present. So you can see here, CD44 percentage. This is a 18 weeks mouse after reconstitution of CD34. We also have a B cell present. So this is great for a B cell depletion study for your immune disease. So the thing for the CD34 compared to PBMC is CD34, they can reconstitution B cell, myeloid cell. But you can say need a longer time for the T cell mature, probably need 12 weeks at least. So need a waiting, not like PBMC, you can, 2 weeks can ready to use. It need a very, very long time compared to PBMC. But and the biocytogen, we have the off-shelf CD34, HSC, BNDGL15 mouse off-shelf. So it's ready to use. The mouse already reached to more than 10 weeks. We have 600 mouse off the shelf every month. So it's great for you, support your research, especially for the B cell depletion. So here is a very, very highly summary, different model.
Hi, Dr. Chen. Sorry to jump in. We're a bit short on time. Maybe we should quickly wrap up before we move to the Q&A right now.
Sure. Thank you. Thank you for the reminder. So we very highly summary is a human gene logging immunization model. We can log in different type of human gene to support your different normal targeting. We can provide a cell service for you. And here is the IVIS study. We do a lot of CAR-T. We have done 100 cases. So we are the CR company. We are well established. We are experienced to do those kind of cell survey study. You can see the LNP distribution for your CAR-T in vivo CAR to screening your vector for your CAR-T, and also for the different readout. We can do Luminex MSD FACS study. And also for the, if you look at solid tumor, you can see the IHC and also immunofluorescence study can show very great CAR present in the solid tumor. I hope can see more and more those data can, CAR-T can solve those solid tumor infiltration issue. And I have one highly summary that biocytogen, we are the humanized mice provider. So we provide the mouse, and also we provide a service. Some differentiation with other companies is we can provide overage. So we cover the overage. For example, if you want to have a 100 mouse in a group, we will provide, for example, 20% more mice, 120 mice. Those are 20 mice we will not charge for you. So you can have a 3,000 mice peak from biocytogen, and also it's 100% guaranteed because mouse from us, we don't need a purchase mouse from other vendors, so it's just one stop. Last but not least, you can go to our website to check on our catalog. If some targeting, we already have the mouse for you. Yes, sorry about the time, and that's it for my presentation. I will back this. I know a lot of people want to have conversation with Marco. I will back this conversation to the Marco. Thank you, everyone.
Thank you, Dr. Chen. And thanks, Dr. Ruella, for those excellent talks. We already got many questions in the Q&A box, so let's go ahead and take a look at those. And to our audience, if you haven't submitted your questions yet, feel free to do so now. All right, let's take the first question here. I think this is for Dr. Ruella. Did you try ketogenic diet in other mouse backgrounds? Oh, Marco, you're muted.
My apologies. We tried the BAP-C. We tried the. I think we did do a model of Black 6 as well, and then we have did the NSG, and we saw that in all these models where it was working.
I think there's a follow-up question on that. Keto diet is very popular. Do you think it can be useful for other indications as well, as it increases T cell viability? Did you try BHB beyond CAR-T?
Well, yeah, that's a great point. I think yes. I think that it's already used, for example, in epilepsy, and in other setting ketogenic diet. I would say that we need more clinical data before recommending the use of BHB, for example, with CAR T-cell therapy. But the idea is to test. There are, for example, preclinical papers that show that BHB can also boost checkpoint inhibitors for solid cancer. So I think we need to be supported by clinical data and before sort of using it routinely. But yeah, the idea is that we would test many settings to see if it works.
Awesome. Thanks, Marco. And the next question here, could fecal transplant in the patients help contrast the resistance?
Wonderful, wonderful, wonderful question. That's one of the things that we're interested in. There are actually, I think, a couple of trials now that are evaluating fecal material transplant for CAR T-cell. There are no results yet, but the idea of improving the quality of the microbiome before CAR T-cell is out there. Definitely interested. There's a trial open already, I think, at MD Anderson, and hopefully we'll see good results as we see with checkpoint inhibition.
Okay, next question here. Question to Dr. Ruella. First, it's really interesting BHB dietary effect was shown only in CAR T group, but not in the control group. Is there any specific mechanism by which CAR T cells, but not endogenous cells or even tumor cells, could preferentially utilize BHB?
That's a fantastic question. So in the model where, the model that I showed in the xenograft model, you don't have endogenous T cell. So the BHB cannot really, there are no T cells to be helped against the tumor. So we were giving human CAR-19 or human controlled T cell that don't have the CAR. So they're not able to recognize the tumor. We're now we were going back to the immunocompetent mouse models where we're trying to look if BHB can potentially also stimulate a intrinsic immune response. Is a very good point. And the expectation, if there is a T cell that is recognizing the tumor cell, our expectation is BHB would boost the metabolism of that effector T cell and therefore enhancing the overall anti-tumor effect. We're getting the data now.
Awesome. And the follow-up question on that, based on promising effect of BHB as a dietary supplementation, do you think adding in BHB during the CAR T cell culture stage could reap the same benefit?
This is a fantastic question, and we have thought about it. We actually done the experiments where we would give BHB during CAR T cell manufacturing and then give the CAR T cell in vivo without BHB. And the results showed that it's actually not efficacious. So it's not enough to expose the T cell during manufacturing to BHB. You really need to have the BHB as a fuel when the T cell enter the tumor microenvironment and fight cancer cell. A very good question.
Thank you so much, Dr. Ruella. Here's another question. In your work with CAR T cells and BHB, did you also test what happens if you inhibit mitochondrial metabolism? For example, OXPHOS blockade. I'm curious whether impairing mitochondrial function alter CAR T expansion or anti-tumor activity, given that BHB is thought to support persistence through metabolic reprogramming.
It's an excellent question. There are indeed inhibitors of OXPHOS, which obviously will have a very broad effect, not only against the CAR T cell, but also against any tissue that uses OXPHOS, which is most of the tissue. So we have thought about that. I can tell you that there is multiple evidence that boosting OXPHOS is good for expansion, but also for memory formation and persistence. So I would imagine that inhibiting OXPHOS would lead to the opposite effect. We haven't done the experiment just because it would be very non-specific, right? Any cell would not like to be inhibited if you use a OXPHOS inhibitor. But I see your point about trying to block the pathway that we think is involved in the increase of function.
Thanks so much, Marco. Okay, here's another question. Were the effects of BHB or other metabolites on CAR T cells verified in immunodeficient mouse models in terms of cytokine profiles like IFNG, IL-10 of inflammatory myeloid or T cell subsets or cell types involved in reducing the effects?
Wonderful question. Yes, we also saw a cytokine increase in our immunodeficient xenograft models. And regarding the question about the changes in the myeloid microenvironment, we're doing now the experiments to see what's the effect of BHB on the, for example, tumor-associated macrophages, Tregs, and so on. We don't have the data yet.
Thank you. I think the next question going to Feng Qian on the mouse model. So what advantages do gene knocking mouse models offer in accessing toxicity, biodistribution, and durability of CAR T responses?
Great question. I think gene logging mouse provide some advantage over those traditional transgenic or wild type model. I think it's from the 4-layer physically expression. So those human gene is insertion in the mouse locus. So expressed is follow the natural regulation. For example, tissue specific and time specific. I just use those CD8 example. It's expressed in the T cell, CD8 T cell, but not expressed. CD8 is not expressed in the, for example, B cell or CD4 cells, and ensure the CAR T binding and activation occur under the real condition. Second is improve the safety assessment. So those are logging mouse allow to evaluation some on-target and off-target tox in the tissue where the human antigen should be expressed. For the biodistribution study, for example, the CD8 mouse express the human CD8, so the CAR T cell or in vivo CAR can track against those physiologically expressed antigen. The protein, which is critical for the predict clinical outcome, de-risk the translational from the animal to human risk. And last but not least is the duration of response. So those logging models support repeat antigen exposure. Those CAR T can demonstrate the CAR T can maintain the function over time, and the price compared to the human immune system reconstitution model is more reliable. The price is lower. So I hope this answer your research question.
Thanks for the detailed answer, Feng Qian. And next question here. Can you elaborate in CAR T for solid tumors? Which models did you use?
I guess this is for me.
Yes, I think so.
We've done several experiments with CAR for solid tumors, and we have relied on different models. I would say mostly xenograft models where we're using, tumor cell lines, for example, pancreatic cancer, ASPC1, or ovarian prostate cancer, and we're using like mesothelin CAR, HER2 CAR. Now, obviously, there are other targets that are pursued, but we also use immunocompetent models. For example, one model of B16 that is expressing CD19 to make it easier, so we can use a product that we know is working. And we've used also some pancreatic KPC model for moderate. So they are suboptimal, but I think for solid cancers is particularly important to use immunocompetent or PDX because cell lines are quite easy to kill and they don't represent the actual clinical scenario.
Awesome. Thank you, Dr. Ruella. And the next question here. Regarding Dr. Ruella's presentation, the microbiota that provided the most CAR T response benefit are not supported by the keto diet, which was the most beneficial diet for CAR T response. In fact, keto reduces microbiota diversity and reduces Firmicutes species. Any explanation there?
No, it's a great question and a great comment. I would say that while we are still studying the effect of ketogenic diet on CAR T cell patients, as I mentioned, we are collecting these tools now. Well, you're definitely right. I think there are also some similarities. For example, in the fact that some of the taxa that we saw in our microbiome study, they are known to generate butyrate, which is a close structure that has a close structure to beta-hydroxybutyrate, and we know that both of them are beneficial for the immune system. So in other words, I think it's going to be a balance between the expansion. I mean, it could restrict diversity, but as long as the ones, for example, the butyrate producing ones are generated, I think that the effect is going to be potentially beneficial. Keep in mind that we're now using directly the metabolite and not the ketogenic diet in patients. So potentially, and again, we're still getting the data out, potentially the effect on the microbiome will be different than the whole ketogenic diet.
Thank you so much. Unfortunately, I think that's all the time we have for questions today. So I'm just gonna wrap up. Okay, so if you have any, follow-up questions or the questions we haven't answered yet, we will address them later and post them on our LinkedIn page. So keep an eye on that. Or
Yeah,
if you would like,
feel free to reach out to me directly. You can find my email, and I'm happy to answer those questions and connect.
of course, thank you so much. Or if you would like to learn more about this topic, feel free to reach out to Marco, Dr. Ruella, feel free to reach out to Dr. Chen, or you can reach out to us through our website at outsmarting.com, or you can connect with us on LinkedIn. We would be happy to keep the conversation going. And just a quick reminder, if you missed any part of today's session, this webinar will be available on demand, and you will be able to request a recording on our website, or you can find it on our YouTube channel. All right, I think that's all from us today. Thank you again for joining, and we look forward to seeing you at our next webinar. Thank you, everyone.
Thank you, Marco. Thank you,
Thank you,
everyone.
guys.